And they continue to groom and remove the hair, and then finally they have lacerations. And at that point, we have to put them to sleep. Okay? So this is very similar to what's called OC spectrum disorder and trichotillomania, where patients essentially compulsively remove body hair, okay? And all body hair. I went to a conference one where there were 1,000 essentially patients out there with trichotillomania. I couldn't tell after I'd interviewed them and they put off their wigs. Okay, completely, all the hair would be gone. So these mice have essentially what we call trichotillomania in mice. Now, one thing that was curious, if you put a normal mouse in the same cage with these mice that overgrew, excessively grew, pathologically grew, they start grooming it. But the way they groom it is opposite. Before I showed you that they were removing the body hair on what we call the tummy side, the ventral side, and now they're removing the hair on the other mouse. It's a normal mouse in the same cage, and they remove what we call the dorsal hair. So this was important because it told us that it's not likely to have something to do with what we call the peripheral nervous system, that is, outside of our brain, because it's not likely to be able to feel an itch on another mouse. Okay, so it's likely to be much more complex and likely to do something with the brain. And in fact, that's where grooming comes from. The pattern and so on is all generated in the brain. And in fact, we know that circuit, and it's called the OCD circuit, and I'll come back to that later. So this told us that it's likely to be something wrong with respect to something going on in the brain. Okay, if that's true, then this gene should be expressed in the brain, and we didn't expect that because Hox genes are normally not expressed up in the front part of the brain, what we call the forebrain. The way that Joy made the mutant, she actually put things into the gene in two places that made the gene a little bit bigger. And this is the RNA made from that gene. So what you can see, this is normal, plus plus means two normal genes, plus minus means one normal gene, one bad gene, and then minus minus means two bad genes. And you can see that when it has one bad gene, one good gene, you have the normal messenger RNA pattern, and then there's the mutant. And then if you have two defective genes, you only have the mutant pad. So that told us, and this was done in the adult and in the forebrain, and that told us that this gene was being expressed in that part of the brain where we did not expect it to be present. And the reason we didn't expect it to be present is that Hox genes, I told you that they're important for specifying what goes on at different parts of your body. And in fact, the genes are all lined up so that all the different parts, if I could look at one particular gene, then I know where it is on the chromosome. It tells me exactly where it's operating in the body. And Hox genes stopped essentially in evolution when you were a crocodile. Okay, and crocodiles essentially have almost no forebrain or midbrain, and completely a hindbrain, and that's where Hox genes essentially stopped in terms of making those particular structures during evolution. So this was a surprise. Here's a Hox gene, and it's actually functioning up in the brain. We didn't expect that. Now, we is a different we. This is another person in the lab, and this is Shatwa Chen. And what we switched is, I told you that Joy was taking infrared movies. And that's very robust. It's very easy to see what the mouse is doing by looking at a camera and looking at the videotape. But it's extremely labor intensive. Imagine taking movies 24 hours at a time and then looking frame by frame to see what that mouse is doing and then quantitating that. So now we switch to automation. And so what we have are platforms that are very sensitive to vibration. Okay? And so no matter what you're doing, you give off a different vibration pattern. Okay? If you're walking around, you have a different vibration than if you're running. You have a different vibration if you're eating or drinking or climbing upside down in the cage and so on. So simply by measuring vibration on a platform, we can tell what that mouse is doing. We can run this 24 hours a day. We have an algorithm that then quantitates this vibration pattern with that vibration pattern. It simply spews out essentially the data, and it's very quantitative. And this involves actually now 30 instead of one set pair. We're looking at 30 mice and seeing what they are. And again, The mutant, the black bars, are spending about twice as much time green as normal. But now this is all quantitative and robust, and therefore we can then do a lot more different kinds of experiments with it. Now I told you that this gene functions in the brain, but the level of activity there is very, very low. So it's very difficult for us to identify which cells in the brain were actually have that gene functioning. Okay? So what we did was a trick, and it's the same trick I previously talked about. Again, the rosa locus expressed in every cell in the body all the time. Okay, so here we have the rosa locus. Here we have GFP from green fluorescent protein from a jellyfish, a stop site in front of it, flanked by LOXP sites. And then we drive it with HOXB8 crete. So wherever HOXB8 functions, then those cells will turn green and stay green. And it's easy for us to identify it in the brain. And there we got a huge surprise. And that's shown here. And here we're looking now at the forebrain and green cells. And here we're looking at the YFP signal. CD11B is a marker for microglia, and they are coincident. So what I'm telling you, microglia are immune cells, and they're the immune cell of the brain. So what I'm telling you is that immune cells in the brain are responsible for controlling behavior. Because if we inactivate this gene, now it has a very strange behavior, okay? Trichotillomania. So this was a real big surprise. However, and so we wanted to look at this more carefully, and we looked to see when do those green cells arrive in the brain. And there's a lot of controversy in the literature as to the source of microglia. There's one school that says they all get in very early in the brain, what in the mouse would be about 6 days of gestation, just after implantation, and that the brain is just starting to form. And the other school says there are actually two sources, one that's early and one that's later from definitive hemopoiesis. So what we see is P2 means 2 days after birth, P14 is 14 days after birth, and P90 is 90 days after birth. And so just after birth, we see it small amoeba-like cells, which are present in what we call the choroid plexus, in the meninges, which is the covering of your brain, and in the ventricular surface, you have several ventricles in your brain, and we just start seeing it in that surface. And then we start seeing spread very quickly. After P14, they reach saturation. From then on, we have the same number of green cells in the brain, and we think these are coming from the bone marrow. Okay? So that's the other school, which said that one is a bone marrow source, and the other says there's only a single source, very early in embryogenesis. And so this data is consistent with that second hypothesis. And further, we have the first marker that differentiates those two cell populations. Now this makes a very strong prediction. First of all, that the gene, if it's in the bone marrow, it has to be expressed in the bone marrow. And so here we're taking blood and separating it out according to different markers, and then in the presence or absence of crete. And then we look at the YFP signal. And so here's a marker that's true for myeloid cells, B cells, T cells. These are lymphoid cells and antibody cells. And what we can see is as soon as we have crete present, we switch all of these cells over, and so now they start fluorescent green. Okay, so that tells us that this gene is expressed very early in hemopoiesis, in making blood cells, and in particularly in stem cells. And we show this also here. These are two common markers for hemopoietic stem cells, SCAL1, c-KIT. This is that population. If we look at this in the presence or absence of crete, here's that population in the absence. Here's the presence of HOXB8 crete, and we shift that whole population over, saying that it's expressed in stem cells. So it's expressed very early in hemopoiesis. Okay? So the next question we ask, well, if that's true, maybe that those cells are affected by the mutation, so we simply counted glial cells in the brain.